Thank, thank you, Brent. Thank you, Derek, for obeying. Okay, here we go. So we're in the middle of this. So we're drawing a sketch here of what's going on in interphase and in mitosis. So interphase has chromatin in the nucleus. In other words, the DNA is kind of unraveled and kind of sprawled all around in the nucleus. Mitosis, though, what do you think is going to be the difference between interphase and mitosis?
It's going to be like not bunched up like that.
Yeah, it's going to be not unraveled like this,
Be wrapped up.
not in its thread-like. It's going to be packaged in things called chromosomes. Yeah. So now in mitosis, you have that DNA. Actually, the nuclear membrane is gone at this point. So in mitosis, you have the nuclear membrane goes away in a phase that we're going to talk about. It's called prophase. The nuclear membrane goes away, and now we have a bunch of chromosomes and there's no nucleus now. Okay. Remember, these are the two main cycles of the cell, of a cell's life. Chromatin, it's unraveled because, well, we're going to talk about that in a second. We've learned a little bit. We haven't gone into it a ton. But do you guys know, we've talked about how, what's made during interphase?
Proteins.
Proteins. Also, there's something else.
Chromatin.
Well, yes. More DNA, right? DNA is duplicated. But what is needed to make proteins?
Amino acids.
Amino acids are. And what's the blueprint for those proteins?
RNA.
RNA, and RNA is based on?
DNA.
DNA. So here, the DNA, how easy would it be to work with something that's all locked up all the time?
Not very easy.
Not very easy, right? So wouldn't it make sense that here it's kind of unpackaged? I like to liken it to if you're going on vacation or something. If you're going on vacation, do you have stuff sprawled about and you just kind of carry it in your hands and kind of take it to where you're going? Or do you pack it nice and neatly? So, well, let's not ask some of these guys over here. But ideally, we're good. Ideally, yeah, that's true. Maybe not.
You've seen our cars.
Okay, so my point is, what is the most efficient way to to separate things? Pack them nice and neatly. Yep, pack them nice and neatly. And so that's why we do that. Here, it's unpacked because we're using it. Once you get to the location you're at, do you want to pack it in there every time all perfectly and stuff? That's a nuisance. You want to go on the roller coasters at Disney World. Where you don't have time to pack the stuff up and away. So here you have it unraveled because you're using it. Here you pack it up because you're moving it. Okay? We'll get more into that later. All right. So when a cell is finished growing, replicating, what?
Duplicating.
What's it replicating? In interphase? DNA. And doing its job, it either dies
Hibernation.
or goes into,
Sleep.
no, not sleep, mitosis. It either dies or goes into mitosis. And we will watch another video here.
Let's dive into this animal cell and see how mitosis works. We pass through the plasma membrane, revealing the nuclear envelope with its pores. There are two centrosomes to the right of the nucleus and a nucleolus with...
I'm going to actually, it's fine. I'll record it.
Then, during prophase, the chromatin is condensing. Each chromosome consists of two chromatids. The nucleolus breaks down and the centrosomes move apart, building the spindle and asters as they go. The disappearance of the nuclear membrane marks the beginning of prometaphase. During prometaphase, the spindle invades the nuclear region. Spindle fibers are forming and breaking down.
Travis is trying to pick my nose.
When kinetochores capture spindle fibers, they stabilize them by giving them something to pull.
Whoa! What are they doing?
The captured chromosomes are pulled to the equatorial plane, and the cell is in metaphase. As we move our camera, you can see the metaphase arrangement on the equatorial plane.
Too cool. Be quiet. Try to pay attention.
The centromeres divide, allowing anaphase to begin. Two processes are going on. The spindle fibers shorten, and the poles move apart. Both processes move the chromosomes toward their destinations.
Please be quiet.
Yes, girls.
We back away to observe the events of telophase. The spindle breaks down, and the chromosomes elongate as the chromatin uncoils. Nuclear envelopes form, and then nucleoli.
So is the gray thing the whole cell, or is it divided cells?
The gray, hold on.
Most of the cells with
Okay, yes. The whole thing is a gray, but let's go back here a little bit.
Breathe interface.
Where's that gray circle now?
Not there.
It's not there. What was that gray thing? The nucleus. So there's a phase where after they're done moving apart, guess what?
Nuclear envelopes form.
The nuclear envelope forms. The nucleus comes back.
A contractile ring causes cytokinesis, leaving two daughter cells, each genetically identical to the parent cell.
Yep.
What happens to like the organelles? Do they just like split up and then
Good question. Yes.
does one get them and one doesn't?
Nope. They get split up among both cells.
So like one will have mitochondria, one will have lysosome.
Nope.
Or do
There are hundreds of mitochondria at least in most cells. So some that's depending on which ones. Sarah, hey, how's it going, Sarah?
Good.
That's good. Yeah. There are a lot of mitochondria in a cell. So they're able to split open. You can see this one. Oh, okay.
Joey, are you sleeping?
Joey.
I had just my eyes closed.
Okay. Can you turn the lights back on, please? It's been a late night, evidently. Okay.
Joey had a rough night last night.
Yeah, I did.
Okay. So all of these organelles are able to split into pieces, okay? So part of the endoplasmic reticulum can go with one cell, part of it can go with the other. It's pretty much the case with everything, okay? Good question. Okay, we are mitochondria song.
Yeah.
Oh, please.
Not
City song.
today.
I did the right song too.
Now, let's. I'll get you. We need to pull these up for the other class anyway. So. Everybody come and get one of these. What's that? Don't get the ones on this side. I'll actually.
Turner, hook me up. Nope. Really?
Yeah,
I got Rick.
I prefer 5 though.
Hey, Travis.
Sleeping.
Travis.
There's everything in the usually expected.
Right. Now, the DNA in mitosis is packed the most. In interphase, it's all threaded apart. That's why it takes up the whole space of the nucleus. Okay? So because it's all threaded loosely, it takes up the whole space of the it takes up space of the nucleus, but there's spacing between those threads, okay? That space is taken up by proteins that are copying that DNA. It's or making RNA from that DNA. Derek, can you set that down, please? Thank you. So it is important for the DNA to be unraveled during interphase so that it can be used. So that's why it's less dense. But when it's in mitosis, it's all bound up tightly and wrapped up so that it can be moved really in an organized fashion. Okay? So most densely packed, mitosis. Packed so it can be moved. In interphase, it's unraveled. Why?
So the proteins can copy.
Yeah, that's one thing. Also, so DNA can be copied. So in other words, it's unraveled so it can be used. Does that make sense?
What was the difference between chromosomes and chromosomes?
No, that's a good question. It's really, chromatin, actually, for our purposes, they have like 2 definitions for chromosome, which is silly. I wish they would be a little more clear. And I've seen it double definitions multiple places. For our purpose right now, we're going to say chromosomes are when they're densely packed and you can see them. Okay? When you can see the kind
What's
of
chromosome?
chromosome is like this. Okay?
Chromatin is when it's all
Chromatin is when it's like this.
So it's the same thing.
It is the same genetic material, only arranged. Yeah, it's the same material. It's still DNA, but it's just this one is DNA that's been packed and put together tightly. Guys, shh, quiet.
Quiet.
Chromosomes are tightly packed chromatin during mitosis.
Yes.
Like noodles and pasta.
Yeah,
Okay.
the chromatin is packed up
Noodles and spaghetti.
and forms chromosomes.
Same thing, but they're different.
So you write the label next to the picture.
Chromatin. Chromosomes.
You label them? I'm labeling them on the side. I know it's on the
We labeled them.
We labeled them on the last slide too.
No, I do. This is tightly packed.
But hey, you can do it again, if that helps. Especially since this is kind of the review section. Actually, this question we just answered. Why might this be beneficial to the cell? Go ahead and write your answer, because we just talked about it and we've kind of written the answer down too on this slide. If you don't know the answer, then that emphasizes even more why you should write this down.
My mitosis is beneficial?
Yeah, why is it beneficial that DNA is densely packed in mitosis? Good. Okay. Yep, so it can be moved. Sorry for the redundancy there. I kind of got ahead of myself a little bit. Does that make sense? Any questions? Thank you for asking those. Good?
You've got this down.
Sweet. Okay, the next thing I want us to do, I just want us to look at a couple animations online so you can open up your computers. We might only get through one, but
Want us to leave
that's
these
fine.
out?
Yeah, you can leave them out because the other class is coming. I suppose this time.
Where's the Sanimation at?
Do go to Google Chrome or whatever your browser. I don't care. Oh, hey, I need to stop this recording.